All right, we're now talking about section 6 for chapter 12 on the nervous system, and we're talking about the plasma membrane of neurons. We want to be able to distinguish between a pump and a channel and describe the 3 specific states of a voltage-gated sodium channel, to list the channels and the pumps that are located along the entire neuron and identify the general function of each. Identify describe the 4 functional neuron segments, and including the distribution of channels and pumps in each. So with pumps, pumps basically need membrane proteins that maintain the concentration gradient by moving the substances against their concentration gradient. They require cellular energy. These neurons, the neurons have sodium potassium pumps and calcium pumps in their membranes. Channels, there are protein pores that are in the membrane, and they allow the ions to move down their concentration gradients into or out of the cell. When we say down the concentration gradient, remember we're always talking about from high to low. When they open, they allow a specific type of ion to diffuse. Leak or passive channels are always open for continuous diffusion. Chemically gated channels are normally closed but open when a neurotransmitter binds. And voltage gated channels are normally closed, but they open when the membrane charge changes. So 3 states of voltage gated sodium channels. When it's in a resting state, the activation gate is closed with an inactivation gate open, and the entry of sodium is then prevented. An activation state, the activation gate is open due to voltage change, and the inactivation gate is open, and sodium then moves through the channel. In an inactivation state, the activation gate is open, inactivation gate is closed, and the entry of sodium is prevented. This only lasts a short time because the channel is quickly reset to resting state. And this little example here, you can see it. So we have the resting versus activation, inactivation, and you can see with the gates that are basically referring to inactivation gates. It's these bigger walls here, and they will move in, as you see in the last slide with inactivation, when the inactivation gates close, whereby the activation gates, this little flap or a little extension here, if you will, and it remains open during activation, of course, and it stays in this open state in inactivation, but it closes during resting state. So in resting state, you got inactivation gate open, activation gate closed between the interstitial fluid and the cytosol at the plasma membrane. And then with activation, you have them both open. And with inactivation, you have the inactivation gates closed and activation gate open. For modality gated channels, they're normally closed, but they're open in response to a stimulus other than a chemical or voltage change. They're found in the membranes of sensory neurons that respond to changes in their environment. Some receptor neurons of the skin have modality-gated channels that open in response to mechanical pressure, as an example. The entire plasma membrane of the neuron has basically leak channels, in this case potassium leak channels and sodium leak channels, and then of course has sodium-potassium pump, and these maintain the resting membrane potential. And as far as a distribution of pumps in the channels, the plasma membrane of the functional segments in a neuron has receptive segment, where the dendrite and the cell body with chemically gated channels, chemically gated chloride channels here. Initial segment, the axon hillock, has voltage-gated sodium channels and voltage-gated potassium channels. The conductive segment, the axon and its branches, have voltage-gated sodium channels and voltage-gated potassium channels. And the transmissive segment at the synaptic knobs have voltage-gated calcium channels and calcium pumps. So here we have a receptive segment with our chemically gated cation, or cation as I always like to jokingly say, or just become a habit of mine, channel, and the chemically gated potassium channel and the chemically gated chloride channel, the receptive segments here for the dendrites. And there's being, of course, cell body and dendrites showing here. At axon hillock, we have the initial segment, the voltage-gated sodium channel and the voltage-gated potassium channel. Then we have the conductive segment where along that axon, where we basically have the voltage-gated sodium channel and voltage-gated potassium channels. And the entire neuron essentially then has the sodium-potassium pump, so basically a sodium leak channel and a potassium leak channel throughout. So here we have basically the distribution of pumps and channels in the plasma membrane of the neuron, showing the transmissive segment here, the synaptic bulbs, where we have a voltage-gated calcium channel and a calcium pump, because calcium, of course, is what's required here. That process, we get the synaptic bulb, when we talk about the synaptic knob meeting up with another neuron, then that junction there. So what did you learn? You should be able to describe the 3 states of voltage-gated sodium channels in the neurons, which functional segment of neuron contains chemically gated channels, and which functional segments contain voltage-gated channels. So for section 7, you should be able to, with the introduction of neuron physiology now, integrate the concepts of voltage, current, and resistance with neuron structure and function, describe the conditions of a neuron at rest, define resting membrane potential and state its typical value for neurons, and explain how the resting membrane potential is established and maintained in neurons. So when we're talking about neurons and Ohm's law, electrical energy is just the movement of charged particles. Neuron activity is dependent upon electrical current. Voltage is potential energy, amount of difference in electrical charge to the two places. It's measured in volts or millivolts. The current is the movement of charged particles across the barrier that separates them, and it can be harnessed to do work. And then resistance is any opposition to the movement of the charged particles, like the barrier. And it's an increase, and it's shown with an increase in resistance to lower the current. Now, Ohm's law, current equals voltage or resistance. Current increases with larger voltage and smaller resistance. And when it's applied to neurons, charged particles are ions, and current is generated when ions diffuse through channels. And voltage exists across the membrane due to unequal distribution of ions, positive and negative between inside the cell and outside the cell, and interstitial fluid and extracellular fluid. The membrane offers resistance to ion flow, and this resistance changes due to the actions of gated channels. And resistance, of course, decreases when the channels open. And the characteristics of resting neurons. Now, ions are unevenly distributed across the plasma membrane due to the action of pumps. The higher concentration of basically the potassium in the cytosol versus interstitial fluid is typically the case, where there's typically a higher concentration of the sodium, the chloride, and calcium in the interstitial fluid, more so than the cytosol. So naturally, of course, they tend to leak across from higher to lower in that sense. Gated channels are closed in the functional segments of the cell. There's an electrical charge difference across that membrane of that cell. The cytosol is relatively negative compared to the interstitial fluid, and the resting membrane potential is typically negative 70 millivolts. The difference can be, you don't have to worry about that exact number, but that's technically true. The difference can be measured with microelectrodes, one inside the cell, one outside, and a voltmeter or voltimeter to measure it. Basically, we have here the chemically gated cation channel here, noting, and then we have our chemically gated potassium channel, chemically gated chloride channel. This is all part of the cell body, right? Of course, our axon hillock here between the axon and the cell body. And this is the receptive segment here, the cell body. The initial segment is where the axon hillock is, essentially. The conductive segment is all on that axon, right? And along that axon, that conductive segment, we have the voltage-gated potassium channel, and of course the resting membrane potential is negative 70 millivolts. We have more than one of these. That's just one. There are multiple voltage-gated potassium channels, and of course we also have our voltage-gated sodium channels along that conductive segment. And the voltage-gated calcium channel, basically in the transmissive segment, where calcium is playing a significant role there. So with resting membrane potential, potassium diffusion is the most important factor in setting resting membrane potential. That's worth noting and remembering. Potassium diffuses out of the cell due to its concentration gradient. It tends to be higher within the cell, and so it's going to go from higher to lower to go outside the cell. And that potassium diffusion out is limited by the electrical gradient, because the pull of the negative resting membrane potential on the positive ion. Because we're talking about typically being negative on the inside and positive on the outside, and that'll play a role in how much is allowed to diffuse across. If potassium were the only ion that leaked, the resting membrane potential would be where the potassium concentration and electrical gradients are at equilibrium, which is basically about negative 90 millivolts. That's where we find equilibrium. Since there are a few sodium leak channels, sodium also influences resting membrane potential. Sodium diffuses in due to its concentration gradient, tends to be higher outside the cell, and the electrical gradient, where it's more negative inside and positive outside. This small sodium leakage means the resting membrane potential is less negative, and that makes it at negative 70 millivolts. And so the role of the sodium-potassium pumps, by pushing 3 positive charges out and pushing only 2, the pump contributes to about negative 3 millivolts of the negative 70 millivolts total. Okay? And more importantly, it maintains that concentration gradient for these ions. And so we have here a neuron at rest with resting membrane potential, and we see interstitial fluid outside, the plasma membrane, the cytosol, and here you can see basically our greater concentration in general of that sodium and the chloride outside the cell, interstitial fluid, and a greater concentration in general of the potassium and the phosphate inside the cell. And so we have our sodium-potassium pump here, and we have our leak channels Here, this being a potassium leak channel, and a sodium potassium pump, another one here. And then, of course, we have our sodium leak channels as well, shown as purple here. And our resting membrane potential at the moment here is negative 70 millivolts. So what's the role of ions? The phospholipid bilayer and the plasma membrane channels and neurons are relative to the concept of current, voltage, and resistance. Describe the conditions of a neuron at rest in terms of the resting membrane potential. The concentration gradients for sodium, potassium, chloride along the entire neuron, and calcium, technically 2+, at the synaptic knob, and the state of the gated channels. Then explain how a resting membrane potential is established and maintained in the neurons. And we'll come back for section 8, the physiological events in the neuron segments here shortly.